Hello everyone, we're here for lesson 7 this week. This is Daniel, me, and Derek for Stephanie. And so we're going to start off with question 1. We're looking at the genomes of different viruses. And so for the first one, we can see that A has 28%, C has 22%, G has 24%, T has 0%, and U has 26%. So since there is no T's and 26% of U's, we know this is RNA. And since the percentages are close, we can tell that it's a double-stranded and consistent to exonuclease. We know that it is circular, not linear, because that goes in and attacks the edges. But if it's circular, you can't attack the edges. So if we go on to virus 2, A is 34%, C is 37%, G is 14%, T is 0%, U is 15%, and it is sensitive to both. So because of that, we know it's RNA because of the U, not the T. Same nuclease. And so if we look at genome for virus 3, A is 20%, C is 30%, G is 32%, T is 18%, and U is 0%. RNA. We know it's DNA together, and it also says that it is sensitive to both, so therefore we know it's linear. And then for virus 4, A is 18%, C is 33%, G is 24%, T is 25%, U is 0%, and it is resistant to exonuclease. And so because of all those things, we know that it's DNA because there's no U. It's single because those numbers are not close together. And it's circular because it is. So the question 2.
Hi. Okay. So, number 2. So, for A, topoisomerase 2 is composed of 4 protein subunits and uses ATP to compact bacterial chromosomes, while a nucleosome contains 146 to 147 base pairs of DNA and 8 histone proteins. B, the DNA double helix is 2 nanometers wide. Adjacent base pairs are separated by 0.34 nanometers in the DNA double helix. C, topoisomerase 1 introduces positive supercoiling into DNA and cuts one DNA strand. D, the origin of replication in E. coli is called ORIC, ORIC, whatever. E, in the Hershey-Chase experiment, bacteriophage T2 proteins were labeled with a radioactive isotope of sulfur 35, while bacteriophage T2 DNA was labeled with a radioactive isotope of phosphorus 32. And then F, the sugar in uridine is ribose, while the sugar in deoxythymidine is deoxyribose. That's 2.
Perfect. All right, so now for question 3. So question 3, part A. So what we have is we have 2 chromosomes, chromosome 14 and chromosome 21. 14 is 109. It's 109. It's 109 million base pairs long, and 21 is 48 million base pairs long. Now, then it says that they translocate, the 2 of them translocate, so they fuse together. Then it says at the end For this problem, assume that the translocation chooses the entirety of chromosome 14 to the entirety of chromosome 21. So I need a different color marker to show this. So what happened was chromosome 21 got its full length, and then chromosome 14 has its full length, and they fused end to end. So for part A, it says how long is the DNA portion of the translocation in meters? So first, we have to establish how long it is in base pairs. So it's 48 plus 109 because they're fused end to end, which is 157 base pairs. 157 million base pairs rather. So then what we do is we take the number of base pairs, 157 million, and multiply it by 0.34 nanometers, because there's that distance between each base pair, and then we have to convert it from nanometers to meters, so we multiply it by 10 raised to the power of -9 meters, and that gives us our answer, which I'm going to put over here. The answer is 0.0538 meters. So that's for part A. Now for part B.
5338.
Oh yes, you're right. Thank you. Yeah. Okay. And then part B. How many nucleosomes are found within this translocation chromosome? Assume that the average length of the linker DNA is 50 base pairs. Okay, so actually, this is that. So for the linker, we're going to show this out first. So what you got is it's a different color actually for this one. So we have this whole chromosome up here. What happens is it comes along, it loops around a nucleosome, and what it does is the nucleosome inside here. What you got is inside the nucleosome is 140. We can do 6 or 7. It's 146 or 7 base pairs that wrap around the nucleosome, rather the histones, and then you have 50 base pairs between each nucleosome. So to find out how many nucleosomes there are, you have to take 146, add it to 50, you get 196. You take the total number of base pairs, which is 157 million, and divide it by that number. So I'll have that up here. So it's 157 million divided by 196 base pairs. Yeah. And that equals, where is it? Yeah, equals 8.01 times. Sorry, we're not in a room here. Times nucleosomes. Okay, part C. How many H2A molecules would you expect to find within this for each? Let's use orange. So inside each nucleosome, there's 2 of each kind of histone. You'll have 2 you see that? Yeah. We have 2 H2As. Hey, it's fun. 2 these. Yeah. So in each nucleosome, there's two H2A histone proteins. So what we've got to do is take the total number, what's the word I'm looking for, of nucleosomes, which is this number. We have to multiply it by 2. And when we do that, we get 1.6. All right. The radio loop domain, we got a whole bunch of DNA and to a protein. Okay. So here's the protein or the protein chain. It's called a scaffolding, and it kind of something like that. Anyway, so you have anywhere from 25,000 to I think it was I don't remember how much it was. The minimum, yeah. The minimum is 25,000 base pairs of DNA will bind to one of these scaffolding proteins. So what I'm going to do is I'm going to take 157 million. Yeah, sorry. 157 million base pairs and divided by 25,000 base pairs. And that's going to give us the total number of radial loops, which is 6,280 radial loops, radial loop domains, yes. And that's it for that one. So that moves on to number 4. I don't know if that's it. I can do it if you guys want me to.
Are you gonna do this?
Oh yeah. Are we ready? Ow. Okay. Okay, so number 4, the bacterium, Mycobacterium tuberculosis, has a single circular chromosome that is 4,410,000 base pairs in length. So for A, if the chromosome straight out linear. So just for A, we just simply just take the 4,410,000 by the 30.meters. Where am I? Yes. To give us 1,499,700 nanometers. And then we want to convert that to micrometers, which gives us 1,499.4 micrometers. So this is our answer for A. For B, we pretty much just take our answer from A, 1,499.4, and then if we want to add that into the circumference formula, so we divide both sides by pi, which means the diameter would be 478. Now moving on to C. So C, suppose the The Mycobacterium tuberculosis genome is organized into loop domains, with each loop domain containing 50,000 base pairs. If one of these loops was stretched out linearly, how long would it be in micrometers? So we take the 50,000, and then again we multiply it by the 0.34, which will give us 17,000 nanometers. So the microscopic chromosome, we take our original count, 4,410,000, and then we divide that by the 197 base pair thing that we saw in question 3. That gives us 22,386. However, now we want to find out how many histones we have, so we multiply this by 8, which gives us 179,086 histones. And that's all she wrote for number 4.
Perfect. I'm gonna switch you. Okay.
Forgot this is a thing.
Cool. So we're gonna go on to number 5. And so for number 5, we're looking at a bunch of different molecules. And so the first question is, which of the molecules in parts A through F are nucleosides? So nucleosides don't have polypeptides connected to them, or not polypeptides.
So phosphorus groups.
Phosphorus group. Sorry, phosphorus groups. So they don't have any phosphorus groups connected to them, where nucleotides do. So if we look at that, the nucleosides are A, E, and F, and then the nucleotides are B, C, and D. So that brings us to the next question. Which of the molecules in parts A through F contain ribose? And so you can tell the difference between a ribose and a deoxyribose, because it'll have, I can draw those. So ribose will have 2 OH groups, or deoxyribose will not have the second OH group. So if we look at our molecules, the ones that contain ribose are C, E, and F. The ones that are deoxyribose are A, B, and D. Which takes us to the next one. Which A through F contain pyrimidates? Have the 2 or it's the 2-ringed, what is that word? Nitrogenous base.
Yeah, that's a word, yes.
Yes, that word. And so the ones that have that are C, D, E, and F. And that is all the questions for this week. Does anyone have anything else to say? Are we good? Okay, have a wonderful weekend.
Hooray.